Hi everyone. We will discuss about the two principal mechanisms of the adaptive immunity against the extracellular bacteria in this video. The first mechanism is the antibody, and the second mechanism is the helper T cell. In order to stimulate these two mechanisms, there are three stages of the process that our immune system has to go through. There are two parts in the stage 1 that occurs at the same time, and then the stage 2 and the stage 3, the production of the antibody. When there are too many bacteria that intruding our body, or when the bacteria is replicating too fast and our innate immune system is not able to eliminate them, then we will depend on these two mechanisms of the adaptive immunity to eliminate those bacteria. In order to start, the bacteria will be engulfed by all these phagocytes. And later on, the phagocytes is going to digest. It is going to digest this bacteria, and then they will extract their antigen and present the antigen on the surface through the MHC class II molecule. So later on, these phagocyte that is presenting the antigen on the MHC class II will act as the antigen presenting cell in order to activate the naive CD4 T cell later. So this is what happened in one part of the stage 1. At the same time, the B cell, the naive B cell, will also attach to one of the bacteria, right? So the B cell receptor on them will recognize the antigen on the bacteria as well. So now we will focus on the first part first, which is on the activation of this naive CD4 T cell that will lead to the production of the helper T cell first. So when the antigen presenting cell is presenting the antigen on the MHC class II molecule, the naive CD4 T cell will come and recognize the antigen with the T cell receptor. Then, at the same time, the CD4 marker of the naive CD4 T cell will also bind to the MHC class II molecule. So with this confirmation, then co-stimulation occurs. So co-stimulation normally occurs between the molecule like the B7 from the APC and also the CD28 of the naive CD4 T cell. So with this co-stimulation, then now this naive CD4 T cell will be fully activated and undergo clonal selection. Then the clonal selection is going to produce the memory helper T cell and also the effector cell helper T cell. So over here, the effector cell will be the one that is going to activate the B cell later. So before entering the stage 2, now we will look at the second part of the stage 1, which is the B cell. So just now we have mentioned that at the same time, the naive B cell will be exposed to the same bacteria. So this same type of bacteria with the same type of antigen will be recognized by the B cell receptor on this naive B cell, and thus the binding of this antigen to the B cell receptor will activate this B cell. When the B cell being activated, the antigen that is being recognized by the B cell receptor just now will be undergo the internalization and then being expressed on the MHC class II molecule of the B cell as well. So the activated B cell will have the bacterial antigen that is being presented on the class II MHC. Then this B cell will then act as the antigen presenting cell to present this antigen to the helper T cell. So now we move to the stage number 2. So in stage number 2, The activated B cell and the activated helper T cell will go and search for each other, and they will come close to each other. Now, the activated B cell will they will act as the antigen presenting cell to present the antigen that is being presented on the MHC class 2 to the helper T cell. Helper T cell is going to use their T cell receptor to recognize the antigen. And at the same time, the helper T cell will have the CD4 marker that is going to bind to the MHC class 2 molecule. So this conformation will activate the helper T cell. So helper T cell is going to release this cytokine. And this cytokine is going to fully activate this activated B cell. Now the B cell becomes fully activated and undergo chronic selection. So when it undergo the chronic selection, it will produce the memory B cell. At the same time, they also produce the plasma cell. So the production of the plasma cell, which is the effector cell of the B cell, means that it will start the production of the antibody. The plasma cell will release the antibody, and this will mean that this is the starting of the stage number 3. All of this antibody, once it's being released, then it can go and bind with the antigen on the surface of this bacteria. So the binding of this antibody to the antigen on the bacteria will cause 3 outcomes. So the first outcome is that it is going to activate the complement system. The antibody binding to the antigen will form an antibody-antigen complex. So this complex will be able to activate one of the complement protein. So when it activates the complement protein, it's going to cause the activation of a cascade or a series of the other complement protein. So all of these complement protein, it can actually lead to enhanced phagocytosis, or it can cause the inflammation, or all of these complement protein can also act together and form the MAC, the membrane attack complex, and create a hole, create a pores at the membrane of this bacteria and cause the lysis of the bacteria. Another outcome is that this complex of this antigen-antibody complex can also lead to opsonization. So meaning that after the antibody bind to the bacteria, then the FC region of the antibody can actually go and bind with the receptor on the phagocyte. So when the antibody bind to the receptor on the phagocyte, then that means that this bacteria cannot run away. And this bacteria will be stuck at the membrane of the this phagocyte and allow the phagocytosis to occur easily. And this is the so-called opsonization and enhanced phagocytosis. And lastly, the last outcome, when the antibody binds to the antigen on the surface of the bacteria, it actually neutralizes the bacteria. So this means that this bacteria is no longer able to attack any other body cell. They will become neutralized and not able to cause any harmful effect. So with that, that is basically what happened in the first mechanism. Next, we'll look at the second mechanism, which is the helper T cell. In fact, the activation of this helper T cell is the same. So the helper T cell that is being activated not just release the cytokine to cause the chronic selection of the plasma cell and then the production of the antibody. This is only one of the functions that this helper T cell can do. So besides producing the cytokine that leads to the production of antibody, the helper T cell can do 2 more things. The helper T cell can actually release a cytokine to induce the local inflammation. This can be done by using the cytokine, release the cytokine to recruit more neutrophil and also the monocytes to the sites of the bacterial infection. So later on, this increasing number of the neutrophil and the monocyte can do the phagocytosis to eliminate those bacteria. At the same time, it can also release the cytokine. The helper T cell can also release the cytokine in order to enhance the phagocytic activities. So meaning that this cytokine will basically make these macrophage or monocyte or neutrophil or this phagocyte to be activated, and then they will actually do the phagocytosis more effectively. So with that, this is basically how the two mechanisms of the adaptive immunity against the extracellular bacteria.